Okay, are we good? All right, welcome everybody to the second session of the Harbor and Estuary Conference. I'm Rob Buchanan, and I'm going to be moderating today's session, Managing Co-Benefits. We're going to be talking about water quality, resiliency, stewardship, and public access projects, and the way that those projects can work together to advance different goals. So we've got we're missing one speaker today. So instead of having 4 speakers, we'll have 3. That's a good thing though, I think, in that we'll have a little more time to hear from each person. And you can see on your screen there our 3 speakers. We've got Elise Meyers, who's just got her PhD at Columbia, works at Lamont Doherty. John Mclaughlin, who's been in the restoration business forever. Well, not quite forever, but for a very long time. And Emma Butzler, who's a student at Meyers College and is going to be talking about her study. So I think that the bios are of all of our speakers are available. And instead of going through them now, I think we ought to just go ahead and proceed with our first speaker. And oh, we've got a poll. All right. So let's do that first. And I'm not quite sure how this works, Rosanna. Maybe you could jump in and explain, but we want to know from you what you're working on, what you're interested in. And these are the choices. I'm not sure how this poll works. Can you explain that?
Yeah, so the choices should be on the right-hand side of your screen where you see Q&A, polls, chat, and community. You should be able to see the poll already live when you click on poll, and we're looking for you to respond right into the question there. And then we welcome everyone to submit questions for the panelists in the Q&A, and then any comments or discussions can be done in the chat.
Okay, I hope that's clear to everybody. And I want to briefly mention the one speaker who couldn't make it today. That's Jennifer Gonzalez, who works with the city of Hoboken, was going to be talking about one of their new resiliency parks in Hoboken, and she was unable to make it because of a last-minute conflict. So I'm sorry about that. So with that, should we wait a bit for the results of the poll to come up, or are we going to present those later, Rosanna?
We can go back to those later. Thanks.
Okay. So we're going to start with Elise Meyers from Lamont Doherty, and she is the title of her talk is Sewage Pollution and Sinking of Pathogens to Better Predict Swimmable Conditions at Varied Locations, including Piermont and Nyack. So Elise, why don't you go ahead and take it away? And I hope this works out.
Sure. Thank you very much for the introduction. Thank you so much for the invitation to be here as well. I'm excited to talk to you today a bit about some of my work that is related to quantifying the impact of particle association on fecal bacteria persistence. If we could advance the slide, please. Thank you. So sewage and other anthropogenic waste contamination of surface waters is an issue of global concern. Contaminated inputs into local water bodies can occur via a wide variety of wastewater sources. So just some images up here. In particular, things like increased precipitation can overwhelm existing infrastructure, leading to sewage inputs into local surface waters from things like overwhelmed wastewater treatment implants, sewer overflows, and also permanent or regular discharges. So it's projected that these sewage contamination events will only increase in frequency and intensity in the future, which makes this issue even more pressing for us to address right now. And some major factors influencing this increase in sewage contamination are increasing urbanization, so having more people concentrated and loading existing infrastructure, also having increased impervious surfaces. Another one is then changing precipitation patterns, so frequency and intensity of precipitation. So you can think of recent hurricanes we've had even in tropical storms in the past year. And then also the fact that we rely on some antiquated and out-of-date infrastructure that is more susceptible to being overwhelmed. So if we want to more effectively predict the increasing impact of sewage pollution on surface waters, that's where we need to think about modeling and trying to understand the dynamics of those systems. And that's what motivates my research on fecal bacteria pollution in the Hudson River. So at the bottom there, you can see this image of a potential discharge, a point source discharge coming out, and then we have people there recreating in this water. And so minimizing that exposure risk is something that motivates my work. I grew up near another urban influenced estuary, the Chesapeake Bay, and so my interest in public health, especially environmental health and inequality, motivated me to pursue this kind of work at the interface of environmental and public, or environmental science and health science. And as was mentioned before, one of my primary field sites is circled here, actually on the right-hand panel, that Hudson River estuary map. This is the lower Hudson River estuary. So Piermont Pier, which is in Piermont, New York, and Nyack, those are my predominant field sites. Next slide, please. Okay, so those sewage contamination outfalls that I mentioned can lead to the discharge of fecal bacteria into surface water. So before we go any further, I wanted to identify what fecal indicator bacteria are for those who maybe don't think about them as much. So an FIB, like Enterococcus, is used to indicate the presence or discharge of sewage in pristine waters. So at the top right, there's this image, which is a Petri dish showing Enterococci colonies from one of my experiments. So there's a gridded filter. It might be a little bit hard to tell, but that's put on top of a Petri dish, which has enriched media. So water is filtered through that gridded filter, the white filter that's there, placed on the media, and then the bacteria are allowed to grow for 24 hours at 41 degrees Celsius. And so then 24 hours later, every Enterococcus cell that went onto that paper now grew into a colony of many cells, which we see as a dot. So each of those dots is many, many cells. And then I can then count each of those colonies to find out how many Enterococcus cells were in the initial water sample. So basically that's just a way to say, this is how I find out how many Enterococcus cells were in an initial sample. So though things like Enterococcus are not pathogenic themselves, and that they largely don't cause illness in humans, they are found to be positively correlated with gastrointestinal illness. Can you forward on the slide, please? Thank you. So in this plot that we're seeing here, so if you look at the x-axis, which is along the bottom there, we can see the abundance of Enterococci increasing to the right. And then on the y-axis, we have the NGI rate, which is the number of gastrointestinal illnesses per 1,000 swimmers. So essentially more people are getting sick, more swimmers are getting sick as we go up that y-axis. So while we see that this is a noisy relationship, it's generally quite positive. So we see that there's a strong relationship between increased Enterococci counts, increased these fecal bacteria counts, as well as increased gastrointestinal illness. And the reason why FIB correlate with gastrointestinal illness is because abundance of FIB correlate with the abundance of pathogenic bacteria. Can you go forward on the slide again? Thank you. So what we have on the plot here, so along the x-axis, again, I'm showing you the concentration of Enterococci cells, but in this plot on the y-axis going up, we're having the concentration of Shigella and Salmonella. So Shigella in black and Salmonella in blue. And so in that upper corner, again, if you look at those pictures, now I have a picture of a Petri dish from some of my samples where I was growing up Salmonella and Shigella. So in this case, again, water was filtered through that gridded filter. Salmonella and Shigella are able to grow on the same type of media. And then in this case, the Salmonella are those black colonies and the Shigella are the colonies that are beige there. But again, what I want to draw your attention to here is that it's a noisy relationship, but we again are seeing this positive relationship. The more fecal bacteria we have, the more Enterococci we have, generally the more Salmonella and Shigella, those bacteria that actually make people sick. So you can kind of think of this like canary in the coal mine type thing, that like the Enterococci are warning us that there is a problem and that the problem would be Shigella or Salmonella. Next slide. And now one more thing I want us to understand before I go into some of the results and the design of my experiments is bacterial particle association. So it's really important to understand this when we're considering how fecal bacteria are transported and they persist in surface waters. And so how I define particle association is my work is purely operational. So that image that you see there with those circles with like the small orange dots, so we can think of the orange dots there as Enterococci cells. Some of them are attached to particles, those yellow circles there. The whole water sample goes into the top of this filter tower here. There's a filter in that kind of bottleneck part of the tower there. So then the bacteria that are attached to particles stay at the top and those that are free living go to the bottom. So that's how I define those two there. And it's a 3 micron pore size filter. Go forward one more, please. Thank you. So extraenteric ecology is described as those ecological dynamics of organisms once they transition from an ideal habitat to a less ideal habitat. So here, the guts of warm-blooded animals like ourselves, fecal bacteria are very happy in there. But then if they go into a water column, that is not where they're happy. And so because of this transition from an ideal to non-ideal habitat, we assume then that extraenteric ecology is dominated by loss or bacterial mortality. Go forward one more. Thank you. So the difference, though, is that particle association has been suggested to be beneficial for bacteria. So particles can be a more favorable environment for bacteria because they increase shading. So thinking about UV disinfection, if the bacteria is on the bottom of the particle, as it is in one of those graphics there, potentially there's some protection from sunlight exposure. There's also potentially localized nutrients on particles, protection from grazing or chemical stresses, and also sinking. And so this then means that particle association could result in longer persisting fecal bacteria pollution. And that makes it really important to interrogate how particles impact extraenteric ecology for more effectively predicting sewage pollution persistence. Next slide. Thank you. So now let's go over the major controlling factors for fecal bacteria persistence that I interrogated in my work. So first we have sinking, which probably only affects the particle-associated bacteria, those that are our circles, dots attached there. Then we also have light exposure, which is known to be the dominant cause of mortality for fecal bacteria from the literature. So thinking again, UV disinfection, like we've all become a bit more familiar with during the pandemic. And then finally, we have dark period or background mortality of the fecal bacteria, because if you remember, they're in a non-ideal habitat. So there's this dark period loss. Next slide. So now going into the data, we're looking at sinking rate data first. So this is data from some experiments by my collaborator, Andrew Jewell. And so in the first plot on the upper portion here, we're seeing time plotted along the x-axis and then the percent remaining in suspension plotted along the y-axis. And what I want you to take from this is that when we're looking at the particle-associated cells, which are the blue line, or represented by the blue line there, they do not remain in suspension over time. So they are actually able to sink, while the free-living cells in yellow are actually remaining in suspension. And in the second plot, we're looking at along the x-axis the sinking rate, so how fast they're sinking, versus on the y-axis the percent with a sinking rate greater than x. And while that's a bit of a complicated way to show this, it's looking at a sinking rate distribution. And so that blue line again, which is our enterococci that are particle associated, they sink with this distribution, and the median, so you know, is about 1 meter per day. So being attached to particles does result in sinking for enterococcus. Next slide. And now we have to think about those dark temperature-dependent growth rates, that background growth rate for those bacteria. So first I want to orient ourselves to the data or to the figures that we're going to show here. So we have increasing temperature. So in the left-hand side there we have 5, and then increasing to 18 on the bottom, and then over to the right is 28 degrees Celsius. Enterococcus is the only one that's shown right now, which is that orange group there. But if you see in the top right-hand corner, I'm going to show you some other data from Salmonella and Shigella, which are those pathogenic bacteria that come from inside human guts. And then I'm also going to show you Vibrio data, which is a marine pathogen. So another pathogen, but that does behave a little bit differently. So it's kind of a nice check. And then particle-associated fractions are denoted by the stripes there. And then overall on the y-axis, we're looking at growth rate. And so if it is above that gray line at the 0, then it is positive growth. If it's below that, then that is mortality or loss, which remember, that's what we're expecting things to be dominated by. Forward one. So then generally what we're seeing now that we have all of this information and context is that with higher temperature, there's higher mortality. And that's especially true for the free living fraction. So the particle associated fraction looks like it's persisting decently well. But as we increase temperature from 5 up to 18 or 28, we see this increase. Next slide. And now if we uncover the rest of the data, first let's focus on Salmonella and Shigella. So that's that dark blue there, and then the green color as well. So we can see that there is largely similar behavior for Enterococcus and for Salmonella. So there's this increasing, when you increase in temperature, there's a decreasing growth or increase in mortality for both Salmonella and Enterococcus. Those are those that are circled in green there. And then if we think more about Shigella, it actually, like, surprisingly behaves more like, so Shigella and Vibrio behave more similar to one another. So we see that actually with increasing temperature, we see increasing growth rates. That is not something we would have expected for an enteric group. So remember, happy inside of human guts, didn't expect it to be happy in these experiments out in the water column. But something that we need to make sure we understand if we're trying to effectively predict these concentrations. Next slide. And then one more thing I wanted to draw your attention to in this is that across all of these different genera, we saw that particle association decreases loss, and then in the cases circled in red, which is quite a few of the cases, particle association actually leads to positive growth, which is highly unexpected. And this challenges that overall assumption that I have been bringing up before, that fecal bacteria dynamics are dominated by loss when they enter surface waters. Next slide. Next slide, please. Thank you. So now let's quickly take a look at the dominant growth rate for light-dependent bacteria. So we know from the literature this is going to be the most dominant loss rate. So along the x-axis, we have our division between free-living and particle-associated. Y-axis, we have our growth rate per day. Can we advance real quick? So what we see from this already, so all of these are negative, they're below 0, they're significantly greater than temperature-dependent growth. We went from 0.5 to 1 per day on the y-axis before to now 0 to -40 per day. Advance one further. Thank you. And then also from this, we're seeing that if we compare the free-living versus the particle-associated, so the left panel versus the right panel, particle association consistently reduced light mortality, 50% to 5 times as much. So it pays a lot to be on a particle. Enterococcus had the greatest light sensitivity, so potentially as an indicator, we need to consider that if we're realizing that our other bacteria are dying at lower rates when exposed to temperature, or exposed to light, sorry. And then noting again here that Shigella and Vibrio rates were significantly different than Enterococci and actually behaved much more similarly, which was very intriguing. Next slide. So just putting this all together real quick, light-dependent growth is dominant as we expected from before. Particle association of fecal bacteria in general, we can now say increases major growth rates. They're more positive. It induces sinking, likely increases persistence. That's actually work that I've done looking at modeling this, and you can see that in the link that I had posted before. And particle association is also an important consideration for water quality management. And finally, there's some more research that's needed on the differences between indicators and indicated pathogens for their extraenteric ecology, because if we're using Enterococcus to predict these other bacteria, it's really good to do this kind of work to ground truth and understand how those bacteria relate to one another, especially when exposed to different perturbations. Last slide. And with that, I would like to thank my collaborators on this project and thank you all for your attention. I look forward to answering questions later.
Great, thank you very much. I would say that that presentation absolutely complicates the picture that I had of testing, of water testing around the estuary, and really adds a significant factor that really haven't thought about before. We always assumed if we took samples from the water that we were getting a picture of the level of contamination. But what you're suggesting is that we've got to look deeper. We've got to kind of literally kick up the sediment to have a true understanding of what's going on and what the risks might be for human contact. So in other words, if a boater or a swimmer is getting in the water and stirs up all these particles by just by walking in the sand, say, you could increase your risk of exposure. Is that fair to say?
So the sediments do act as a reservoir for a lot of fecal bacteria, and there has been work looking at that in the Hudson and other places as well. In the case of the bacteria I've been working on, I've been taking water samples from the surface water. So the bacteria are actually coming into the system, particle attached and free living. So there's both entering the system that way. And there's quite a few particles in the Hudson, as many people here know. So this is something that we do need to think about because of that prevalence of particle association for bacteria in the Hudson.
I hope we can come back to that at the end of the hour and think a little bit more about what that's going to mean for regulatory agencies and just for the general public, because I think this is another step in trying to understand the whole picture of water quality.
Absolutely.
So thank you very much, and let's keep rolling. But I would be really interested to hear what people have. And I can't see, just so those of you who are watching this out there know, I can't see your questions and won't be able to until the end of the presentations. But at that point, we definitely want to review them and pass them on to the panelists. So let's move on to John Mclaughlin. His title is Managing Director of the Office of Ecosystem Services, Green Infrastructure and Research. That covers a lot of ground with the New York City Department of Environmental Protection, or the DEP. He's the Bureau's Director's Senior Advisor on the design and implementation of citywide ecosystem restoration and innovative ecological projects for habitat and water quality improvement. He's done a lot of really cool stuff over the years. And I think he's going to talk about a couple new things right now. But John, why don't you go ahead and take it away?
I know. Thank you, Rob. Sometimes it does feel like I've been doing this forever on some days, not every day, but some days. And Elise, that was a great presentation, and I think it's a good tie-in to what I'm about to say. I mean, folks know that New York City region is an ultra-urban watershed, where many of the natural systems have been replaced by concrete buildings, roadways, you name it. So a lot has changed within the watershed, that some things are irreparably changed, and it's hard to bring them back. But that doesn't mean we don't stop trying. It just means we need to think a little outside the box. Over the last 20 years, I mean, DEP has invested about 15 billion to improve water quality, and I think some of those dollars are paying off when folks are talking about placing oysters in the harbor. You see whale sightings, dolphin sightings. So there's a lot of benefit that's beginning to show from that investment. But now we're at that point where we made a very large investment, and trying to capture that last piece is rather elusive, because it gets to the knee of the curve where you could keep on spending $1 billion, but you get a 1 or 2% change. So we need to look at other potential solutions for that. So if we go to the next slide. Yeah, so as I mentioned, we have two research efforts currently underway for improving water quality, and it's specific to removing pathogens from the water body using natural systems. In this case, it's using rib mussels, Buchanan, Temissa, and using tidal wetlands. Freshwater wetlands have been studied extensively for their water quality improvements, both in terms of nutrients and also pathogens. But tidal wetlands, not so much. And the reason for that is it's a little hard to monitor a system that's ebbing and flowing, twice a day, and there's many different inputs that get in there. But we hope to begin to quantify some of these numbers with these research efforts. Next slide. Yeah, so this is just some background, literature that search that was done, by others, and the way wetlands, tidal wetlands in particular, that's all I'm talking about, not freshwater, but tidal, that the pathogens are removed, through sedimentation, adsorption, interception, and predation. And these are just some of the studies that have looked at FIB. And to your point, Rob, you just mentioned that the sediments are a sink for FIB, and they can be up to 1,000 times higher than what's in the overhead water column. That's a generic statement. I don't want to say that's applicable everywhere, but that's just one from this one study. But the FIB densities can diminish tremendously, tremendous, by just being subject to daylight. The wetland system that we created, you'll see in the upcoming slide, for some reason, and it's unknown, but it's the first 10 meters of a tidal channel that seem to do the greatest loss for bacteria on either side of when the tide's coming in, on either side of that channel, it's about 10 meters on either side. So that's why the goal was to increase the channel ratio, to increase that that proportion that's subject to degradation. And the wetland system that was built, will monitor on an incoming tide and an outgoing tide. The system is kind of isolated, so it's at the top end, of Alley Creek. So there's only one inlet, so it's very easy to measure the incoming, and then we can also measure on an outgoing tide what those concentrations are, because it's also feasible that wetlands, from an avian source, are also introducing pathogens to the system. There's been a couple of papers that indicate it could be in some locations up to 40% avian. So even if we removed all man-made introductions, avian would still be a major source. And if one thinks that New York City has lost 85% of its tidal wetlands over the last 150 years, just picture all those wetlands with bird juice. There's another 220,000 acres. But then also, looking at the water quality as it comes in, it's also looking at the sediments to see what gets sequestered there, and also what is desiccated, or UV degradation, or just soil microbes, other soil microbes that break down the bacteria. That will be a monitoring period for about 2 years. It's anticipated to begin in August 22. We're pulling together the contract now. It will be with the Science and Resiliency Institute at Jamaica Bay. So it has many academic partners, Stony Brook, Cornell. We can tap into Rutgers and other academic institutions for this information. Next. And this is the wetland that was built at Alley Creek. I don't know if folks are very familiar geographically, but this is in the close to the Douglason area. The water body at the top of the picture, that's Little Neck Bay, and the tracks running through the middle there are the Long Island Railroad. The wetland system on the top is natural, and you can see it has these meandering channels in it, not straight channels. The wetland on the bottom is the one that was created to have these sinuous channels. So we've been doing wetland restoration for a number of years, and we typically want to optimize low marsh coverage. So you just bring in one main channel and then have maybe a 10-on-1 slope to maximize your low marsh coverage. But that's not how a natural wetland system would appear, as evidenced by the top image. But the whole point of this research, as the water's coming in, you see on the bottom, tide going in and out of the system, 10 meters on either side of the channel. That's when the most predation occurs. The sinuous channels create greater contact. The bacteria attach to the vegetation, and that's how you get desiccation and UV degradation. And Elise pointed this out about the UV, the importance of UV in doing that. So that does help quite a bit. Next. And this is just an aerial image. The larger wetland on the bottom was built in 2010 as part of our CSO. That was the Alley Creek CSO. That little line you kind of see going through the dark green is just a pathway that leads to the outfall. And beneath that is a 5 million gallon tank that stores the sewage, while it's raining, and then when the rain is stopped, it's fed back to the wastewater treatment plant. But you can see in that design that we did in 2010, and there's nothing wrong with this design. It's just its goal was different. The goal was to maximize low marsh coverage. So we just made one channel that brings the tide in, and then submerges the wetland vegetation. The image on the right is the new wetland that was just completed, and you can see it has the primary channels, secondary and tertiary channels, and that's all how a more natural wetland system would be. And all along those channels, you can see the one main channel, we can place water quality sensors and take grab samples as the tide is coming in to get a sense of what's in the water. And as it goes through the wetland system up to the top of the tertiary channels, there'll also be other equipment there, and also taking additional grab samples. So we can compare the two, both on an incoming and an outgoing tide. Next. Okay, and then on the other one, I know there's been a lot of attention to oysters, and they are the iconic species that once dominated the harbor. But rib mussels may be their other companion that can also help in water quality. As I mentioned, we're working with SRI, and Cornell and Stony Brook to develop this work. This was submitted as part of our long-term control plan to the state for Jamaica Bay. We're looking at these natural systems in lieu of disinfection, chlorine, because that has its own set of potential issues with chlorine byproducts as they react with organic material in the water. So we're trying to look at these other natural systems that, in addition to removing pathogens, will also then provide habitat. So the rib mussels can filter, down to, the 2 micron range, which and a little bit lower. That's approximately in the bacterial size range. So they're very good at doing that, and just their gill structure is also is designed not necessarily for bacteria, but the way it is designed just makes it a very good vacuum, so to speak, in terms of capturing bacteria. And these are just some previous studies that were done, showing the capture efficiency. And we've done our own microcosm experiments, and right now we're in some mesocosm experiments, but the microcosm experiments have shown similar capture rates, around 85 to 90%. So that's, it was nice to know we were consistent with previous research. Next. And this is kind of busy, but we had to look at, since the bacteria concentrations are highest, during a CSO event, we had to look at various parameters that would change within the water column and what that means to the rib mussel, whether it's actively feeding or closes up, or whatever the case may be. So initially we looked at salinity. If you look at the data, based on the time scale, salinity can drop very quickly. And so in the lab, we subjected the mussels to an immediate drop of salinity from, say 25 parts per thousand down to 0. And basically they stay alive, but they clam up, so to speak. But in reality, in the field, it's more of a gradual change over a period of 2 hours or so. It goes down and then rebounds, rather quickly. So we try to replicate that in the field, in the lab, and you'd see even when salinity drops very low, there's still the efficiency. I should have said, by the way, we're looking at Entero and E. coli. In this case, the blue is E. coli and the yellow is Entero. That even when salinity drops very low, down to like 5 parts per thousand or lower, there's still like a 20%, 15% efficiency. And that's only for a short period of time. Once they rebound back up, the magic threshold number appears to be 12.5 parts per thousand. That's where they tend to, not be so impacted. So we would place, when we do this full scale, we would place these out of an area that's not subject to a constant low salinity. And then
John, just a couple minutes left. Wanted to give you a heads up.
I'm going quick. No, the one on the right is the temperature. Those pulses you see, those rises, those are where we actually inject additional bacteria. So we had to know all the mussels responding to the bacteria, and clearly, you start off with a certain concentration. Over time, levels drop. We throw in another dose. The mussels take over, and the same thing happens. And we looked at 3 different temperatures. 15 degrees C, 21, and 28 C, because we're primarily looking at the recreational season. And those would be the water temperatures one would tend to get between April and say October. 28, not every year, but it's possible some years you could get up to 28. So there's no difference between uptake of Entero or E. coli during those different temperatures. We're looking at different mussel sizes. This is a mid-size of a mussel, and again, generally, there's the uptake is still very good, no matter what the size. But obviously, the larger the mussel, the greater the uptake. But it was rather insignificant once you got above the 55 millimeters. And then turbidity, obviously a big one. I mean, during a CSO event, water's going to get a little turbid. Again, they didn't clam up as much as we thought. And again, it's a temporary thing. The turbidity is high for a period of time, then drops, and then they refeed. And then the big thing of all this, it doesn't matter if the mussels take all this in, take the bacteria in, under all these parameters. The question is, what happens when it goes through the system, and through the pseudofeces and the feces, and generally, if it's not consumed through the metabolism of the mussel, a large fraction of the E. coli and Entero emitted in the pseudofeces and the feces were dead. So the mussels it's not just a pass through, it actually is, it is a lethal dose for the bacteria. Next, and I think that's just one more slide. Yeah, then what we're doing next is, these were all on the microcosm level. Now we're building tanks in the lab with a flow through system as opposed to a beaker. And it's testing density and the spatial arrangement. And then once we get the information from that, we intend to put this in the field in Bergen Basin, in Jamaica Bay, and test it in situ for a number of years to see how it actually works on a much larger scale. But so far, all the data done to date is very positive, and I think that's it. Sorry, I went over.
No, that's fine. That's fine. So we're really talking about two projects, one in Alley Creek on the sound side, and the other in Jamaica Bay. The one on Alley Creek is complete in terms of the construction of those new wetlands.
That is correct.
And the Bergen Basin project, the mussels project, is you're in the kind of preliminary design phase, and as soon as the permitting and the lab portion is done, and you're satisfied that it could work, you'll try it. Is there an obstacle in growing these ribbed mussels? Is that going to be a difficult thing?
Yes, and that's why we're reaching out to the hatcheries sooner rather than later. I mean, it's not we're using the rib mussel, so it's not commercially available. But if given a economic incentive, to grow these, there's no reason why a hatchery wouldn't set aside some space to do the growing for us. But we do realize that is that is a major obstacle that we hope to have perhaps 20 to 30,000 mussels, roughly in the 50 millimeter size or larger by next September. And then the following year, hopefully another 20 or 30,000 in the field. The large scale implementation is over a period of 4 or 5 years. So, we estimate currently that we may need up to 40 million mussels. So that's why it's going to require a tremendous effort in getting hatcheries, to grow this for us. But if the product, if they know the product's going to be sold, then it shouldn't be that hard. It's just a question of getting them to grow it. Unlike oysters, which may produce millions of spat each spawning time, rib mussels are on the order of several 100,000 at each spawning. So it is a order of magnitude lower.
Less prolific. One huge advantage from the regulator standpoint is that nobody is going to be tempted to eat these rib mussels because why?
Right. Well, I've never eaten one, and actually I don't eat mussels anyway, but from what I understand, rib mussels have a extremely bad taste. You know, folks eat the blue mussels. Rib mussels do not, for the most part, are not a human consumption issue. So that attractive nuisance issue tends to go away. Which and we've had conversations with the state, over the last 3 years. So it's not as if we're trying this and then hoping the state buys into it. We've had conversations over the last 3 years, and they are extremely interested in seeing the additional research that we're doing.
I think we might want to ask Paul Greenberg about his recipe ideas before we go any further to make sure. But okay, that's fantastic. 2 projects to think about, maybe in some senses to watch and sort of weigh against each other, or as in collaboration with each other. But that should be very interesting to see those results in the next few years. Let's I'm sure there'll be a lot of questions about that, but let's move on and hear from Emma Butzler, who is a senior at Meyers College studying environmental science and economics. And she's also the recipient of a Polgar Fellowship from the Hudson River Foundation. And so she wants to tell us about a river that probably very few people know about or even know where it is, called the Fecal. So Emma, why don't you take over and tell us where that river is? Is it river or creek? I'm not even sure. And tell us why. Okay, go ahead.
Hello, everybody. My name is Emma Butzler, and today I'm going to talk about my summer research experience, some of the people I met, and how this small tributary is an integral part of people's lives. So a little bit about myself. I'm a senior at Meyers College, and I am studying environmental science with a minor in economics. And I grew up in central Pennsylvania, where natural resources are abundant, which drove me to pursue environmental science and focus on some of the environmental issues. Next slide. So over the past year, with the help of my amazing advisor, Zion Kloss, and funding from the Hudson River Foundation, I designed and carried out an individual research project based out of a Hudson River tributary. My project examined nitrate variation in relation to decentralized septic systems in the Fecal watershed, which this is a map of the Fecal watershed, and it's located in Dutchess County, New York. The study was a socio-environmental approach to addressing excess nitrogen from faulty infrastructure that ultimately reached the Hudson River and affected the Hudson River in forms of harmful algal blooms. And the stream is very unique because it starts in rural forested areas, then flows through residential Hyde Park, and it becomes an urban stream as it flows through the center of Poughkeepsie before discharging into the Hudson River. Next slide. So my methods for my project were relatively simple. Every 50 meters, I take a YSI nitrate measurement, and then every 150 meters, I take a water sample. So I mapped out 50 meters on Google Maps, and the idea was just to walk up the stream and collect data. I ended up collecting 400 data points, which is equivalent to walking 12 miles of this Fecal stream. Next slide. So on the very first day of my research project, I didn't really know what to expect. I ended up scaling a waterfall. I ended up going through 2 tunnels that had absolutely no light, and you couldn't even see the end in sight. And I ended up falling 5 times and getting my waders completely drenched. So there were so many aspects of this project that I wasn't prepared for at all. But one of the aspects that I didn't consider at all was, and that made my project super special, was the people that I met along the way. Next slide. So like I mentioned before, the stream is unique because it flows through a diverse range of areas. It flows in the city of Poughkeepsie, so it flows next to parks, apartments, businesses, and then upstream it flows past people's backyards. And the stream has a lot of character. It turns into waterfalls, ponds, lakes, and wetlands. And what I realized through this experience was Dutchess County residents really take full advantage of this stream. As you can imagine, two people dressed in these really ugly waders carrying bright orange dry bags and a cooler and having really, really loud mouths were bound to get people's attention. So I was able to collect stories and perspectives from people all throughout this small watershed. So today I'm going to share some of those stories. Next slide. So I met Stacy in the center of Poughkeepsie, and I met her twice actually, because I walked the city section a few times, and I met her from her window with her cat. And Stacy's apartment overlooks the Fecal on one side, and then on the other side overlooks the busy 3-lane highway, well, not highway, road that goes through the center of Poughkeepsie. Stacy expressed just how staring out the window was kind of an escape from the city setting. She was able to hear the water flowing, see the wildlife. For her, this stream provided her with peace. The stream is a home to a lot of wildlife, but it's also a home to Aaron. I met Aaron under a bridge in the center of Poughkeepsie as well, and his home consisted of a mattress, some makeshift curtains, blankets, and a suitcase. Aaron didn't really speak much about his personal life. However, he was super excited about improving the Fecal water quality, and he wanted to help clean up the garbage in the creek. This encounter was really moving to me because even though it seemed like what he had was very little and he didn't obviously have great living conditions, he was still invested in some environmental issues. So for Aaron, this stream was a home. I also met Luke within the city of Poughkeepsie, and Luke took full advantage of the proximity to the stream. He used the stream to water his home garden in his backyard. Luke grew onions, peppers, and green beans. And one of the things that really stood out to me was Luke used this garden as a way to get his kids involved in gardening as well. So for Luke, this stream provided him with the capacity to have his own garden and fresh vegetables. The last person I'm going to talk about is Garrick, and I met Garrick upstream a little bit in residential Hyde Park. After sharing my research project, Garrick shared his childhood experience with the stream. Garrick reminisced about playing games with his little brother and growing up with the stream in his backyard. Decades later, Garrick's children now play in the stream. So for Garrick, the stream fostered him and his children's imagination. Next slide. So although I met quite a few people who shared their stories, most of my perspective on how this stream fit into people's lives came from my own observation and interpretation. Everything was kind of left up to my imagination. So for example, a porch swing that was attached to an old farmhouse that was nestled within the woods facing this dammed up Fecal stream that turned into a lake led me to imagine an old couple sitting there listening to early morning birds as they drank their tea. The playground that was near the stream that had children's toys scattering the banks led me to imagine kids running around on a hot summer day in swimsuits. And then the gazebo that was just yards away from the stream with dangling lights led me to imagine a young couple throwing backyard cookouts that led straight into the night. Next slide. The stream was also a place of curiosity and exploration. I was able to take a group of high schoolers through the stream for a research project this summer. This program was based out of the Carey Institute, and the students were able to experience Poughkeepsie in a whole new way. They were able to understand some of the environmental issues that this urban stream faced. So for the high schoolers, the stream was a place to learn. Next slide. So this resource and waterways just like this all over the country are often tied to people's careers. They have a huge role in the economy. So for example, farming, real estate values, recreation, and tourism. But my project opened my eyes to how these waterways and natural resources play into people's everyday lives. Everyone I met had a very unique perspective and experience with this stream. And I realized that this stream was really integrated into everyone's everyday life. So I want to thank everyone on this panel and everyone that really devotes their career to improving water resources. and protecting our waterways, even if it is a small tributary like the Fecal watershed, because it has reaching impacts on people's lives that often go unspoken and unseen. And I'm really happy that I was able to experience that this summer. So thank you. And are there any questions?
Thanks, Emma. I'm sure there are a lot of questions. I want to start by asking, after this study, I'm curious about where you see your own career going. I assume you're probably going to stay in this field, but I'd like to know if you see a particular focus developing as a result of what you found this summer. I guess I'm saying, are you going to become a social scientist or are you going to stay in the natural sciences?
That's a great question that I have a lot of discussions with my parents about, and they're watching this right now, so I'm sure they're excited to hear the answer again. But I do want to stay in water resources, water quality, and I do like the interdisciplinary approach to water resources. I don't know what the future holds for me. I'm deciding to take a gap year and then applying to grad school as well. So definitely staying within water resources though.
Great. One other question is about the 400 data points that you did collect, and I assume those are the results of your nitrate sampling. And did you test for other parameters besides that? I might have missed that detail.
Yeah, so I tested for nitrate levels and then other forms of nitrate as well as phosphorus. And the phosphorus was tied to the water samples that I collected and then was sent to a lab down in New York City. So I just got those results back too.
And what's your overall, if you had to do a brief sort of summary of the water quality health of the Fecal, where would you rate it?
So the Fecal is considered a Class C stream, so it's not extremely healthy. And I went through and did enterococci tests at various locations, and all of them showed up with pretty high levels of enterococci. So I know Elise and John discussed enterococci quite a bit. It's not a very healthy stream, but it has potential and it's on its way to become a healthy stream.
Great. Okay, well, fantastic work, and and also brave work to just walk up the stream. I'm sure there were a couple of moments where people came out of their houses and said, "Wait a minute, what are you doing here?" But you survived all of those encounters.
Yeah, there were a few moments that did happen, but most of the people were very excited that I was doing this project, and I did knock on everyone's door to access the stream. So I did ask for permission.
Okay, good. That makes sense. Good practice. All right, I've got a couple of questions I'm going to ask. I'd like the panelists to think about questions they might want to ask each other. So we'll do a bit of that, and then I want to turn to a number of questions that I'm seeing posted now in the chat box that I can see. So for Elise, I just want to go back to that question of the regulating agencies, and if you see, or what you see as the next steps for them in terms of assessing water quality based on some of the research that you and your colleagues are starting to do, the connection with particles and how that might alter official monitoring practices or assessment practices. Do you see any changes coming there? Okay. Elise maybe has stepped out of the room, and so maybe I should turn to you, John, and I'll pass along a question from Chris Boyd here. He says, has DEP performed speciation of the E. coli and other pathogens found in the tidal wetlands to evaluate the source of pathogens? So you were saying that some estimates are that up to 40% of pathogens in a tidal wetlands may be bird related. Have you done those studies yet? Are you going to do them? How is that going to proceed?
No, I mean that exists to do that speciation, but it's very costly, and at the end of the day, we do know well, no, it's not the only source, but combined sewer overflows are a contributor to that load. Yeah, so that's why we're focusing on that for the most part. But again, as I mentioned, we could get it down to zero from man-made sources, but from natural sources that still may exist. When you think about when, New York City, when Henry Hudson came up well, it wasn't the Hudson, but when he came up that water body, he saw a very different view than what folks would see today. And there weren't very many folks, swimming in various water bodies where, it would have been an issue. So it's because of the population and the density of the population that it becomes an issue. Not to say that prior to that it was not. It's just the sensitive receptors aren't there.
Okay. Let me just check and see if, Elise, are you back live with us or are you still offline? Okay, because we've got a couple questions about sediment transport and resuspension that I think would be good for you to answer. Okay. Here's a question about rib mussels. And I have also noticed rib mussels in great numbers in some of the more polluted tributaries, Gowanus Canal and Newtown Creek in particular, places where you wouldn't expect to find that much life, and you see them in large numbers. Ira Gershenhorn is asking, I expected to find rib mussels in the Harlem River at Swindler Cove. I did not. I've been told the raccoons may be to blame. Would like to hear your opinion.
Well, I can't speak to the raccoons, but yeah, I mean, there needs to be nearby populations and sufficient nearby populations, and not just one, but multiple populations that would put enough spat in the water column for that to be present. One slide I didn't show, we also subjected the mussels to low DO because again, these are they are pretty tolerant of pretty harsh environmental conditions, and putting them to 1 milligram per liter of DO, 3 and 6, there was very little difference in feeding throughout that range. So they can tolerate some pretty low DO, but in terms of why they're not there, it could be, I mean, predation. I don't know about the raccoons and mussels, but maybe the populations nearby, or the water currents are too swift, or I'm not entirely sure. There could be any number of reasons.
But that relative hardiness would be an explanation of why they are in Newtown Creek and the far reaches of Newtown Creek and in the Gowanus Canal as well. They're hardy, and I mean, one milligram of oxygen is not very much.
No.
So, okay, well, that seems another encouraging thing about them is that they're survivors, and
I should add that not that we haven't given up, but in terms of recruitment, we know within Jamaica Bay, Jamaica Bay has the highest density of rib mussels pretty much anywhere, and there's a reason for that. There's not a lack of food within the bay itself. There's a lot of nitrogen and algae that can get in there and feed that growth, but that's the highest density. You may have upwards of 10,000 mussels per square meter, which is a tremendous density. But the important thing here is they do recruit. With opposed to oysters, not in everywhere, but at least in Jamaica Bay, that question eludes us as to recruitment and the reasons why. And rib mussels also live about 20 years. Maybe others who know more about oysters can correct me, but I thought oysters are somewhere in the 6 to 8 year range.
Yeah, I don't know the answer to that. I've heard different things about it. There was that monster oyster that people said was 40-year-old, but I don't know how they knew that. So,
Gotcha.
Okay, thank you, Elise. Since I'm glad you're back. You had it sounds like a domestic emergency, but you've resolved it. Well done. I A couple of questions for you. One is that just the question I raised earlier about changing regulatory agency monitoring or assessment practices based on your research and the research that your colleagues are doing. What do you see happening? What would you like to see happen in that department?
That's a great question. So something I think that would be really helpful is kind of shifting our standards to look more at enterococcus, which is our brackish indicator. So right now we still are using, fecal coliforms, but we get a much cleaner view if we look at enterococcus for the brackish portion of the Hudson River. And then of course, E. coli is very useful in the freshwater portion. So I feel like adjusting to make sure that we're following the the most effective standards nationwide, I think was like a really good baseline step for us to get to. But then something else for the work that I've been doing, I think that would be really useful to do is to consider how we are potentially putting in particle-associated bacteria or thinking about those basically different methods. So yes, we could have turbulent motion that's stirring things up from the bottom, but also potentially if we're having our overflow events or discharges, if we're able to do more capturing of particle-associated bacteria in that time period, that could potentially reduce the persistence timescale of bacteria afterwards. So thinking a little bit more from like an ecological and holistic perspective of like what are the ways that we can change some of the inputs that we have into the system, particularly because we have an antiquated sewer system that is going to have issues with loading partially because of climate change. So adjusting to make sure that we can minimize the impact or reduce that persistence timescale, I feel like is really important.
So would that mean filters or wetlands constructed at each CSO, or how? I'm just trying to imagine how we would intervene with particle-bound bacteria at the outfall.
Yeah, I mean, I think that something like filters could be really useful. In a perfectly treated, so if something goes through all of those different steps in a wastewater treatment plant, the particle association rate of bacteria can be quite low. And the amount of particles that are coming out of those kind of facilities is very, very low because there is that settling chamber that's available there. So potentially increasing the capacity for a lot of the existing infrastructure would be really important. Of course, that requires a lot of capital investment. So then potentially having something like constructed wetlands that are able to do a bit more trapping and binding of those sediments could be really useful.
I once saw a proposal for barges that would be positioned at outfalls and have wetlands on them. And I think there was just a tidal range issue. At high tide, it wouldn't work, but at low tide, it might work really well.
No, we've looked at that in a very cursory way, but if I can just get back to something Elise said, the wastewater treatment plants themselves are an insignificant source of bacteria because the effluent is disinfected before discharge. It's CSO discharges that would be the primary contributor, and the issue with these things is a question of scale. I mean, New York City uses 1.4 billion gallons of water a day. So, and that same volume of water is treated. So even the smallest wastewater treatment plant puts out, 40 million gallons per day. So yeah, putting a filter, something that's passing through tens of millions of gallons will quickly degrade. So again, it's not saying we stop. It's going from the lab to the field to the in situ that's the big leap. How do you do that? And there's no one solution. It's a lot of things in aggregate. You do more wetlands, you do rib mussels, you do upgrades, water conservation, green infrastructure that the city is doing. It's all these little things that can have the sewer system do what the sewer system was intended to do. But we did an analysis of impervious cover in the city. I mean, on average, it's 72% impervious. And 1 acre of impervious cover produces about 30,000 gallons. And New York City is about 242 square miles.
Yeah.
And if you do 72% impervious, just do the math for Ida when it rained 3 inches in 1 hour. That's a tremendous volume. It's scale. Scale is the problem. So.
Yeah. Well, we are the biggest city in the country, and it's, yeah.
I did want to add, sorry, I absolutely agree with John. I think those are fantastic points to bring up. I did want to mention that for the field site that I'm working at, and it's particularly looking at partially treated discharges. So not necessarily just CSOs, because I think that is oftentimes what we think of, which is really important and very much a critical thing that's degrading our infrastructure or our ecosystems, but also when wastewater treatment plants are overwhelmed, that discharge of partially treated or untreated sewage is also problematic. And so in Orangetown, like the Orangetown sewage treatment plant, like that's more where I'm looking at in Piermont and Nyack. So it's not CSOs there, but we're having that other type of input that is still important to consider.
And Elise, I'd like to bring you in on our conversation.
Yeah, oh, I love that. No, your project sounds so fascinating. I'm glad that we are on the same panel.
Great. Elise, let me ask you one other question that came through, and that's about the from Coney Island Creek. There's from the earlier session today, I think there's a very vocal advocacy community there, and they're concerned about the new ferry terminal that's being constructed. And so one of the early concerns was about sediment being being displaced by the dredging for the ferry terminal and the kind of continued turbulence that ferry traffic will call. And so the question, will the considerable turbulence caused by ferries transiting a narrow channel and then idling at the dock? And I have noticed the way that ferries run constantly to kind of nose into the dock. Will that cause an increase in the toxicity? That may not be the right word, but in the contamination, let's say contamination levels of existing pathogens in the waterway. So that's the question.
Thank you for that question. So I think that so it would be hard to know exactly that, yes, this is what will happen, or no, this is not what will happen, until there's some samples that are taken, just to be sure, because I think that would relate to the depth wherever the channel was, and then based in like generally, okay, how intense are the motors going, like how deep is the turbulence then going down through the water column from this ferry in particular. But turbulence in general can definitely kick up sediments. And I think someone who is more of like a like bed shear stress and fluid dynamics modeler, like for that particular interface, may be really useful. I do mostly like water column turbulent motion stuff there. I will say though that the pieces are all there. So yes, there can be mobilization from the sediments. There's been work that's looking at like if you wade through water like on like the shores near Piermont Pier, like do you kick up a lot of fecal bacteria? Yes. And then like there is more turbulence with the ferry. So like the pieces are there. I would think that there should be maybe some samples that are taken. The good thing is that fecal bacteria samples are relatively easy to collect. So that would be something that I would definitely encourage the advocacy groups to get in touch with scientists about. There are some even like at-home kits that are available for that. But something else that could be useful to think about there would be like chemical mobilization as well. So the lingo that I would use for that, for having turbulence, there would be thinking about the abundance of those bacteria in the upper water column. I think that's the way to phrase it instead of the toxicity of the existing pathogens, because then we're thinking about if they're in the surface waters, that's where people who are boating or swimming or fishing, that's where they're going to interact with the water. If it's really deep down and we only really have like the bottom layer that's being mobilized and it's kind of staying in the lower portion there, it's not great, but people are not getting in contact with that and probably not going to actually lead to gastrointestinal illness.
Great, thanks. Okay, I'd like to go back and I want to ask a couple of questions that are in the chat, but sort of frame them as questions about communication and response from the public that all of you have experienced. So Emma, maybe we could start with you, and maybe you could talk a little bit about your project and the way, and I think you did get some press coverage up in Poughkeepsie, but also I'd like to hear more about how you think the project was received and the kind of follow-on consequences of it. And maybe the way to talk about it is to talk about the group of school kids that you worked with that time and what possible repercussions that's going to have down the road.
Yeah, so one of the ways I decided I wanted to communicate my research and get some of the stories out there was through my Meyers Circle newspaper. I wrote a little article about some of the stories and what this research and waterway meant to the community. And like you mentioned, I did take the high schoolers through the stream, which was very impactful for them because they got to go through the urban setting. In the urban setting, obviously urban streams have a lot of issues, but specifically the Fallkill stream deals with a lot of trash issues. So we saw lots of mattresses, bicycles, just random debris, wheelchairs, blankets, you name it. It was probably in the stream. And we also went through the section with the high schoolers that had the homeless guy. So that was also very impactful for them. But one of the other ways that I decided I wanted to take this research was because I am a senior, I'm doing a capstone project. And I've decided that I wanted to design my own capstone project focused on the trash in the Fallkill. So this is going to happen in the spring, where I spatially analyze and quantify some of the hotspots of trash within the Fallkill. And then the second part of the capping project is the community outreach project. And the goal for that is to get as many community members, Marist students, local high schoolers, Vassar kids, Bard kids, I know they're kind of far away, but as many people as possible into this community project, because I think there's some appreciation when you get your hands in this resource and you're kind of realizing like what this resource is for people. And I also want like everyone to like getting a diverse range of people together to clean up a resource, I think is also impactful, but it will lead to a more appreciation of the waterway. So that's how I'm trying to take this research.
That sounds great. And so maybe you can present next year on the results of that collaborative cleanup of the Fallkill. Can have some before and after pictures. Okay. Elise, what about you? I did notice that you shared a link on the Whova platform of a video that did a pretty good job, I thought, of explaining the whole idea of particle attachment for pathogens. And I'm wondering what other just basically how much success have you had in communicating this general issue to the public, and where your successes and failures have been?
Yeah, that's a great question. So I do try to do a lot of communication, like science communication work and outreach. And the video is definitely one of those efforts to try and make sure that the information that I'm finding is getting out to people in a more accessible way. This is everyone's river. And I think that people can be really invested in these areas. And I think people in general are. Like when I work at Piermont Pier, I'm able to interact with people there, which is fantastic. So then I have people who are helping me take the readings for temperature, or helping me collect a sample. They're talking to me about how when they were younger, this is what it was like. And so getting people involved and making sure that we as scientists are keeping a space for them and making sure that they understand that their voices are valid in our projects and in our research, I think is a really, really critical way to do work that is so influenced by an urban area. And so I usually do a good amount of like scientific talks, those kind of interactions on the pier. But then something that I was most excited by was an activity at the Lamont Doherty Open House a couple of years ago. Of course, now we don't have it in person because of the pandemic. But before, we had about, I think it was 250 kids came through our exhibit. And it was kind of like Hudson River Clue. And so we had them going through the whole system, trying to understand where this water sample, their mystery water sample, came from. And in that process, they learned about what are some of the things that are endangering the Hudson River, and then what kind of creatures live there, what kind of resources do we get from the Hudson, to really make it concrete to people, this incredible ecosystem that's in their backyard. Because a lot of times we think of Hudson as, something that's in the way between New York and New Jersey if you were trying to get over one of the bridges or in the tunnel, and it's so much more than that. So I feel like those, just kind of taking a step back and trying to make that effort is a big part of what I do.
Great. And John, I want to ask you about communications too, because the DEP is I mean, it's just what's your perspective on DEP and how the public sees the DEP and how your projects have maybe altered that perception. I'm just wondering how you see that.
Oh, I can say many of the environmental groups are clearly in favor of these types of proposals. I mean, disinfection, is one of is one of the things that is a big sore topic in the with the environs. So, finding a natural system that can remove the bacteria without chlorine, is a win-win. So in terms of that, we're looked upon favorably, but at the end of the day, we're this big utility that, and I don't say this flippantly, but I always say when I speak in person, can I get a show of hands? Who doesn't have indoor plumbing? So, we all contribute to it, and it has to go somewhere. If you think back in the day when it just went out raw. So again, is it an ideal solution? No, but it's better than what it was. I mean, the water quality in the harbor is the cleanest it's been in 100 years. I mean, ships used to love to come to New York Harbor in the 40s and 50s and 60s because they would get a free scraping of their hull.
In other words, everything growing on the outside of the hull would die because the
water would just
yeah.
fall off. So now that's not the case. The marine borer that's impacting a lot of the wooden pilings. That organism, its predator, hasn't quite caught up in terms of the balance. So they're pretty much having a field day, so to speak. So it's a massive undertaking. Elise pointed out that it is an aging infrastructure. If you saw pictures of the wastewater treatment plants when they were built, there was farmland around the sewage treatment plants. So they weren't in the community. However, over time, the community has come to the door, to the walls of the plants. So it's not as if you can totally redo the plant because you have space. Right now it's built right up to the property edge.
Yeah.
If you were in Iowa, you could build a whole new plant next to it and then 10 years later flick a switch, but that's not the case here. Very difficult, very challenging.
We just have to decommission a few highways and then there'll be plenty of room for more sewage treatment.
Well, that's out of my topic.
Okay, great. Rosanna has reminded me that we did take a poll early on at the beginning of this session. And so if you're able to broadcast those results, Rosanna, I don't know how to do that, but maybe you do. It might be interesting to see who's on the line, and also any last questions that people want to ask, we will pass them along.
I did see 2 questions in the chat. Oh.
Wow, that looks like a dead heat amongst all these categories, right? Oh, I see. Well, the winner seems to be community engagement, outreach, and education. All right. Rosanna, any further analysis there? You're muted.
No further analysis. It's just interesting to see where everyone's at.
Okay. Yeah,
They were
John.
able to select more than one, so. That's
true. That we didn't limit it that way. Okay. Yeah, John, if you want to respond, if you see any particular question that I missed, why don't you go ahead? Tell us what it is. And
yeah, one was from Judith, and she was asking, did I actually say 10,000 mussels per square meter? And the answer is yes.
Wow.
Now, I'm not saying that's the norm, but there are certain areas within Jamaica Bay that can have up to 10,000 mussels per square meter.
Okay.
Then another question was looking at sediment on wetlands for sea level rise, and that has actually been done in Jamaica Bay. It's called thin layer spraying. Like before the wetland totally degrades from sea level rise and other mechanisms, you apply a thin layer of material over the marsh. You know, it's got to be light. You can't put too much, otherwise you smother the vegetation. But it's this incremental application. And you know, it has been done in Jamaica Bay. It was something called Big Egg Marsh that was done by the National Park Service, I believe back in the late 2000s. I'm not sure the exact year. It could have been like 2006 or 7. I'm not sure, but no, that was very successful. So it has been done.
Okay. And for Elise, I think just to kind of summarize these couple of questions about survival times typically for Enterococcus, but I think maybe in general for pathogens that are normal or typically found in sewage, what is a kind of a par time for, and I'm sure it's different obviously for free floating and particle bound pathogens, but in terms of the public, what should we generally think of as a typical kind of clearing time for these pathogens? Are we talking days, hours? What are we thinking?
Yeah, that's a fantastic question. So this goes to kind of like what John was saying before, is thinking about like our lab to in situ types of comparisons. So my results predominantly come from the lab, but then also doing some observations with Riverkeeper, we're able to do that generally in about 3 days after rainstorms, which is usually when we have those really big inputs of fecal bacteria. That's when the water quality goes back down to a level that is within EPA compliance. So then you can think of like a couple days within like 3 days after a rain event, then we should see pretty safe water quality once again. And so that's, you know, related to multiple things. That's related to those major growth rates that I mentioned before, also to sinking, to dilution, to mixing down deeper in the water column via turbulence, all sorts of factors like that. If you had like a really clear water column, so if you're looking in some place like Lake Tahoe, so that was mentioned, I believe, in someone's question. So a very clear water column, yeah, you would expect to see faster mortality of the bacteria because you would have that greater light exposure. Same thing if the water column is more still versus if you have a lot more motion and it's moving down. And then I've done some simulations to look at what that difference in timescale could be. And so the simulations point to having, you know, on the order of like 5 hours or so in like a perfect system where the bacteria stay kind of floating at the surface. There's not much movement. It's clear water. But then, you know, days, much more days longer for more turbulent water or more turbid water, which in the Hudson, we have both of those conditions. So thinking about those two different types of end members there. But definitely it's a good thing for having more experiments to test that in situ to make sure that that range is about what we're seeing. And especially thinking about what's the area that's most important. Like it's most important in that surface water section where we're thinking about human recreation if we're going for water quality, public health type work.
Okay, thank you very much. I think we've all got our work cut out for us. On the other hand, it's a perfect business plan. We've all got plenty of work to do, and there will be plenty of work to do for the foreseeable future on all of our fronts. So Emma, John, and Elise, I want to thank you all for participating. And I want to also, Rosanna has reminded me that we do have a couple of sub-sessions. One, what Piles Creek taught us about pollution response in estuarine animals. That's Judith Weiss, her sub-session, and then City Island Oyster Reef with Sally Page Connolly. So those are the two sub-sessions of this particular panel. And I'm going to ask Rosanna to share her screen and remind us of what's coming up for tomorrow in the conference. Thank you very much. Wait, you're muted. You're still muted there.
Thanks, Rob. Thank you everyone again for attending today's session. I just wanted to quickly remind you all of what's happening tomorrow here on HUVA. Please join us at 9 a.m. for the Safe Space Commitment launch, followed by our 11 o'clock Living with Water. We'll have another training happening at 2 p.m. called Bystander Intervention in Public Spaces, and we'll close tomorrow with an old story made new at 4 p.m. So thank you everyone again for sticking around, and have a great rest of your evening.
All right. Thanks, Rosanna. Thanks, everybody. Bye-bye.
All right, bye-bye.
Thanks so much for the invitation.
Thank
It's an honor to be with
you.
all of you.